What I thought I'd do is just talk a few minutes about the context and environment that we operate in Cambridge and how Abcam fits into that. Tell you a little bit about our growth and what we're excited about in the future, and then come back to what the CBC, I think, offers and why we're excited about that move. And I suspect you, like me, sitting there listening to Sir Hugh and to Andy, were struck by how many times you heard something new and exciting. And we have the privilege of seeing worldwide everything that's going on in biology. And our perspective is this is definitely the most exciting time in life sciences ever. The sequencing of the genome, which was big news now, it seems like almost 20 years ago, was the beginning of asking a lot of questions about biology. And what's happened is the cost of sequencing has gone down. Computing power has become abundant and almost zero cost. Gene editing is now making it possible to be precise in asking questions of biology. And all of those tools are bringing together fields of genomics, epigenetics, proteomics, metabolomics in a way that's creating the most exciting parallel universes of tremendous advances ever. And Cambridge is an exciting leader in that field. And so in that context, I'll tell you a little bit more about one particular area. And if you heard Sir Hugh talking many times, he was talking about proteins. I'm going to zero in on a tool that was developed here in Cambridge to help analyze proteins, and that's antibodies. Cambridge is the epicenter, ground zero of inventing the antibody market. And Cesar Milstein and George Kohler, who were at the LMB, worked very hard in the early to mid-70s to develop the tool that has unlocked not only the research tools that are used to study proteins, but an industry that's now worth $100 billion a year worldwide. And it didn't stop there, of course, even though their work was defined in its timeline, and they were recognized with a Nobel Prize, one of the many in your inventory, in 1984. But then additional work that took place over a long period of time was to make antibodies recombinant and also to humanize those, which then led to the first drug, Humira, which was an antibody to TNF-alpha and helped patients with rheumatoid arthritis. All of that invented here. And in fact, Andy, I think, was an early part of that at CAT. Our part of this is we operate in the 1% of that $100 billion a year market, providing the tools for researchers to analyze proteins and study them, but certainly with an eye towards the applications in diagnostics and therapeutics. And in 1998, Abcam was born. The photo you see here is of Jonathan Milner, our founder, a postdoctoral student at the Gurdon Institute. And in this epicenter of the antibody world, what he felt was missing was an ease of sharing information and data about great antibodies to study proteins. In his case, he was trying to study BRCA2 and to advance our understanding of breast cancer and treatments for that. And he provided, I think, really a breakthrough idea of making it easier for researchers to find the products that they were going to need to accelerate their research. Now, today, Abcam is a different company. I think our aim is to be the most influential life science company for researchers worldwide. We think of ourselves as Tenzing to Hillary, that our customers are trying to summit great mountains and do amazing things. And our job is not to just provide the oxygen, but to provide them with the advice about making the right steps to get to the top and to get them there as quickly and safely as possible. And as such, over the period, we're now the #1 provider and brand for research antibodies worldwide. We're extending our capabilities into clinical applications through collaborations. We're about 1,100 employees. Roughly a third of those are here in Cambridge. And we're listed on the London Stock Exchange at about £2.3 billion. But all those things pale in comparison, or at least the kind of the results of the focus of all of us on helping research scientists advance their work faster than they might otherwise. And that's the mission and purpose of our company. This is a global story. Whilst we started here in the UK and in Cambridge, our business today reflects the mix of life science research. About 5% of our business is in the UK, 95% outside, and the two biggest markets are the United States and the ever quickly growing China. Over 600,000 researchers worldwide are using our products every month, and that results in about 30,000 publications a year using our tools. One of the things we talk a lot about is giving scientists back their Friday night. Certainly when I was a graduate student, I wasted many Friday nights that I would have otherwise productively used in the pub because experiments weren't working quite the way they were supposed to, or I didn't quite have the tools I needed. And conceptually, what we're trying to do is to use the data that we have as a company about what all researchers are trying to achieve worldwide to derive the distinctive insights that were required to invent the products that are going to be needed next, or to invent something that's going to be better to make scientists more productive. And the way that we're doing that is combining a number of technologies and tools that we have in-house. So, for example, we make about 11,500 recombinant antibodies that have been highly validated, but also collaborate with others who are providing them for us, such that we have about 130,000 products available to scientists 24 hours a day, 7 days a week, everywhere in the world. That gives us great responsibility and great pride in having that helping hand in research. As I said earlier, we are extending our work and our capabilities into clinical applications. The way we're doing that is as a partner developing antibodies or affinity binders for applications that go to clinic. I've listed 3 here, but there are many. We made the antibody that's currently being used to diagnose patients that will be treated with Opdivo. And for those of you who don't know, this is a drug that's being used for treating cancer patients, particularly lung cancers. About 25% of lung cancer patients may respond to this type of drug, and what the antibody is doing is identifying those patient subpopulations. They came to us because we're good at engineering antibodies. We do thousands of them every year. We have the kind of scale and ability to design and validate those that is hard to come by in the world. And, but all the development of the regulatory approvals and marketing those have been taking place with both Bristol Myers Squibb and with Dako. In the second example, we're working with a small biotech company in San Diego called Abcam Biosciences. They're about to come out with a prostate cancer diagnostic. We will have made the androgen receptor V7 antibody for that application. And we've had a long-standing relationship with the Michael J. Fox Foundation, creating the tools required for studying how Parkinson's disease works in clinical trials and applications for providing diagnostics and therapeutics in that market. As I said, this is an important evolution of Abcam as we built the capabilities of making our own products, such that that's about half our business today is unique products that are made at Abcam. Finally, let me just finish with coming to the Cambridge Biomedical Campus. For us, I think we see three themes for our home. The first was really influenced by a meeting I had with Sir Hugh before we had decided to come to the CBC, and I asked him, how important is it that we have proximity? In other words, is it important that Abcam's here in Cambridge for you as a research center? And it wasn't clear to us at that time, because we set up at the Cambridge Science Park today, and I can tell you that at that time we felt our relationships with universities and collaborations was stronger outside of the UK than it was here. And the central question we were asking ourselves about the CBC was, would being that much closer help us forge better relationships to serve and integrate ourselves with science? And we felt particularly strong that the answer was yes, but I think that conversation helped crystallize for us what being at the CBC would be like. Absolutely want to continue with our existing catalog to serve and influence the quality of the science that's being done here, because it's a magnificent resource and great research is happening. But also, secondly, to collaborate with some of the new areas where we don't have an existing product and there's a clinical opportunity. We're definitely doing more of that, and having more companies and organizations with that purpose is exciting for us. Finally, we're looking forward to enjoying the site. We're in a number of tired old buildings. We don't get the opportunity to be in one place, and what's being built for us here is a terrific building. But also enjoying this center as a great place to work. If you ask employees at Abcam at the Science Park today, what's it like working there? Many of them are jealous of their colleagues in Cambridge and in Massachusetts, or in Shanghai, or in other parts of the world where we have facilities. because those are buzzing, humming, great centers of innovation and entrepreneurship and science, and we don't have that up on the north side. So the vision of the CBC, delivering a great place to work that has transport links, that's a great environment, is very exciting for us, and we're looking forward to enjoying that. So thank you all for your attention. I don't know if you want to take questions now, but I'm happy to.